compared with healthy people. But we don't know whether, in most cases, we don't know whether it is because I'm diseased, so I have a different microbiota, or it is because I have a different microbiota so that I become diseased. So chicken or egg, which comes first? So that's why we propose that we should still follow Cocker's postulates and trying to answer the causality question. But we need to modify, we need to adapt to two things unique for microbiome. First, the microbiome participation into health and disease should be a polymicrobial nature. So it should not be one single bacteria do everything. It's most likely will be a group of bacteria working on our health or disease. Second, it's not a one-way street. It's not like infectious disease. Bacteria always cause problem. It's actually some bacteria may be beneficial, others may be detrimental or pathogenic. So we need first to do microbiome-wide association study, try to capture all the variations of all the members during disease progression or during intervention or during cross-sectional comparison, and identify all the members which are positively or negatively associated with the disease phenotype. And then we should isolate the associated members either into pure culture or into consortium with defined membership.
Excuse me, Dr. Zhao? I apologize for interrupting. Were you going to share your screen with the slides?
I already did. Sorry.
Oh, okay.
I do not see it.
Yeah.
Oh, sorry. Let me. Oh.
Yeah,
there we go.
Yeah.
Sorry for that.
No worries.
Okay.
No worries.
So basically, after you isolate the associated members into pure culture or into a consortium, but with defined membership, you should put them into germ-free models, germ-free mice, germ-free rats, germ-free piglets, germ-free non-human primates, and then give the environmental conditions, see if you can reproduce the disease. If you can, now you have a autobacterial model of a human disease, and then move to molecular study, try to elucidate the molecular chain of causation from colonizing the gut, producing some bioactive molecule, interact with the host cell, and eventually trigger the disease reaction. So after you finish all of this, you may be able to claim that you have identified the key members of the gut microbiota by producing what kind of effective molecule they can aggravate, induce, or elevate a particular human disease or a disease phenotype. And then they can become biomarker, they can become drug target. So this is the level of rigor we must reach if we would like to move the whole field forward. The approach we use is to basically doing interventional studies by changing diet to change the gut microbiota, and then to see if we can change the disease phenotype and hopefully improving the disease. So before, during, after intervention, we collect urine blood fecal sample, analyzing molecular level variations of DNA, protein, metabolites in those three windows, and correlate them from each other, and also correlate them with the health phenotype, and eventually identify key members or candidates of the key members. So we need this is the overall approach, and but there are three things we need to keep in mind. One is bacterial functions are strain specific. So by definition, strains in the same bacterial species can have up to 30% genomic difference from each other. Human and mice only have 10% genomic difference. So this gives us an idea how different bacteria can be genetically and functionally, even though they are in the same species. So that's why you need to try to go down to species level if you want to identify the truly important bacteria. Second, members in the macro ecosystem, they are not independent from each other. Different members can actually form a group called functional guild. Members in the same guild, they increase or decrease together. And so we need to know what kind of bacteria work together and to form a functional group called guild in the gut microbiota. We also know in macro ecosystems, not all the members are equally important. And for example, in the closed forest, The tall tree species are the foundation species. They need to be there, also need to be above a certain abundance level to be able to close the system and make it structured and stable. If you lose the tall tree species, you eventually lose everything. So they are the foundation species. Then the question is, among the gut bacteria, are there any bacteria who also work as the foundation species for keeping the whole gut microbiota stable? We also need to identify that. So I would like to use our work on metabolic diseases, particularly obesity and type 2 diabetes, as an example, and to show how we may use those conceptual framework and methodology, try to understand microbiome in human health and diseases. We know we have a world epidemic of obesity and type 2 diabetes. And currently, we also know that people with obesity, type 2 diabetes, and they are the hardest group. And they actually have much higher hospitalization rate and mortality rate. And what's wrong? And why so many people start to develop obesity and type 2 diabetes in just a few decades? And many people say this cannot be genetic, so it could be dietary pattern change. And there are many hypotheses. Maybe you have too much fat. Maybe you have too much easy carbohydrates. But our hypothesis is, instead of having too much something bad, we're actually having too little of something essential. That is a dietary fiber. So if you look at the results analyzing the fossil field feces of ancient human, we realize that they had 200 grams to 400 grams of dietary fiber intake every day. But the average American can only have 15 grams on a daily basis. So over eons, our gut microbiota and ourselves are co-evolving under a high fiber dietary context, but now it's changed dramatically just in the past few decades or no more than 100 years. And in traditional Chinese medicine, food has been used as medicine. If you look at those food, they are rich in non-digestible polysaccharides, and you can also call them dietary fibers. We also collected the fecal sample from 314 healthy young Chinese, 7 ethnic groups, half men, half women, and half rural, half urban lifestyle from 18 to 35 years old. And we sequenced their microbiome, and they showed, not surprisingly, ethnic dependent segregation of the microbiome. So different ethnic background associated with a different microbiota, even though they are all healthy. However, if you look at the composition, you found that about 9 genera shared by everybody, regardless of the ethnic and geographical location and lifestyle or gender. And they amount to about half of the sequence. All these 9 genera are short-term fatty producing bacteria. So basically, this is an indication that healthy young people's gut microbiota is enriched with fiber-utilizing gut bacteria. And this is a Lancet systematic review paper commissioned by WHO. They compiled data from 185 prospective studies with 135 million person years of data, and also data from 58 clinical trials, interventional trials, with 4,635 adult participants. All this huge amount of data on low fiber and high fiber intervention or diet intake gave one general conclusion. There's a 15 to 30% decrease in all-cause and cardiovascular related mortality and instance of coronary heart disease, type 2 diabetes, colon cancer between the low fiber and high fiber groups. So this is an indication that dietary fiber supplementation seems to be beneficial to many patients across different diseases. But why? Why it's so fundamentally beneficial? Well, the mechanism probably could only be through changing the gut microbiota, because dietary fibers are non-digestible carbohydrates. They cannot do much to our own physiology, metabolism, but they can be fermented as energy source by many gut bacteria, and then release short-chain fatty acids as side products for bacteria, but they become essential nutrients to human host. There are hundreds of papers published on how short-chain fatty acids provide energy to our colonocytes, mitigate inflammation, increase satiety, and so on and so forth. However, there's a common pattern emerging on microbiota research related with type 2 diabetes, colon cancer, and many other chronic diseases. That is, there is generally a reduced abundance and diversity of short-chain fatty acid producing bacteria, and also the amount of short-chain fatty acids in these diseased people's gut microbiota. And so based on all this, we developed a dietary intervention scheme where we keep macro and micronutrients balanced for the host, and but at the same time include as much and as diverse dietary fibers we can in the dietary scheme. We call it a WTP diet because it's based on whole grains, some traditional Chinese medicinal food, and soluble prebiotics. So I myself was the first volunteer using this diet, make myself lose about 45 pounds of body weight and recover from metabolic syndrome and remain healthy since 10 years ago. And so this was featured as a news article in Science in 2012. If you are interested, you may have a look. So I would like to briefly go over two studies. One is on a genetic form of obesity in children called Prader-Willi syndrome. And they were born with a very low muscle tone, but after weaning, they quickly start to develop hyperphagia and always hungry for food, and very quickly develop into morbid obesity, like 5 years old, 100 pounds. And this is a very difficult form of obesity to control. However, when we are doing a hospitalized dietary intervention program targeting the gut microbiota among morbidly obese children in Guangdong Women and Children's Hospital, we found that 1/3 of the participants actually PWS patients. And they surprisingly responded very well to the intervention. For example, this particular boy, 14 years old, 140 kilograms at the initial, at baseline, and BMI over 60. And he was in the hospital for 285 days. Everybody else finished at, stopped at 3 months, but we let him continue for 285 days. And he was 83.6 kilograms when he left the hospital. And he continued the intervention at home. After 430 days, he was 73 kilograms. So he lost about half of his initial body weight and recovered completely from the metabolic syndrome he had before. And but with no exercise, just the dietary intervention targeting the gut microbiota. Then the question is, it is because the diet, when you change the diet, you reduce the calorie intake for sure. And is it because they reduce calorie intake to reduce body weight and regain all the metabolic health? Or is there all this has anything to do with the gut microbiota? So we transplanted the gut microbiota from the same individual before and after the intervention to germ-free mice. The pre-intervention gut microbiota, all red-colored data, showed that the recipient mice receiving the pre-intervention gut microbiota developed the inflammation in liver and gut in the first 2 weeks. After that, they start to excessively accumulate fat. And but the intervention, the post-intervention microbiota from the same individual didn't induce such changes, and the recipient mice grow and develop normal. So this is the indication that because of the self-controlled study design, and we cannot attribute the clinical benefits completely to gut microbiota. And we also cannot assess how much contribution maybe from gut microbiota, how much from just the low-calorie diet itself, high-fiber, low-calorie diet itself. However, we can at least have some evidence to show that the gut microbiota changed by the dietary fiber probably do something to help. So in order to have a closer look, understand the variations and the response of the major members of the gut microbiota to the intervention, we collected a fecal sample from all the individuals at each time point every month. And PWS patients, they had their gut microbiota. They had the intervention for 3 months. So they each have four sample, and we have a simple obesity group, and they stay in the hospital for 1 month. Each had two time point fecal sample. So in total, we have 110 fecal sample, and 109 worked. 1 sample didn't work. So we have metagenomic data from 109 sample. What we did is we use Illumina HiSeq platform, generated about 80 million reads from each sample. And each read is 150 base pair long. So in general, we have about 10 giga base pair data for each fecal DNA sample. And after we assemble them into context, we recognized a little bit over 2 million non-redundant microbial genes. We have only 109 samples, but we have over 2 million measurements, 2 million variables. This is a huge data set. We needed to do dimensionality reduction. Otherwise, we cannot identify patterns. So we follow 2 biological principles to do the dimensionality reduction. First, even though we have 2 million individual genes, but we know they are not independent from each other. So genes originally coming from the same genome must behave exactly in the same way, because that's genetics. If you want to reproduce, you need to copy every single gene, give that to your offspring. So this is determined by genetics. So all the genes which are originally coming from the same genome would give you exactly the same abundance changes across all kinds of samples. If you see not exactly the same, that's because experimental error. So we set up a co-abundance analysis with a very high correlation efficiency. And then eventually we identified a little bit over 20,000 co-abundant gene groups. Each such group of genes is potentially a genome or part of a genome. And then because we are very much interested in bacteria, so we look for co-abundant gene groups which contain more than 700 genes. And we end up with 376 such CACs. And the total amount of sequence, these 376 bacterial genomes, is already over 70% of the total sequence we generated. So this is the indication that even though we have fungi, we have virus, phage, we have parasites, and we have all the other types of microorganisms in addition to just the bacteria, but gut bacteria are still the most predominant. And also they are potentially the most metabolically active. And also myself is a bacteriologist. So we just focus on these 376 bacterial genomes. And many of these genomes are still just show up in 1 or 2 individuals or just a few samples. So it's then we should focus on the bacteria which show up in more than 20% of the samples, because they are prevalent enough for us to do analysis. And among the 161 prevalent bacteria, we assembled 118 high-quality drafted genomes. More than 95% of genes can be identified from such genomes. So now you have reduced the variable number from 2 million to 20,000 to 376, and now to 161 genomes. So among the genomes, how can we further reduce the dimensionality? Now we apply the ecological principle, because no bacteria actually work independently in the ecosystem. So they should work together with someone else. If they work with someone else, they show co-abundant behavior. So we did a co-abundant analysis among the genomes, and then we deleted them into 18 co-abundant genome groups. And each such group, so each circle is 1 genome, the size of the circle is the average abundance of that genome. The same colored genome, they are in the same co-abundant genome group, so they are potentially a guild. If they are connected with the red line, they also co-occur. And but if they are connected with the blue line, they co-exclude from each other. And now we have 161 genomes clustered into 18 guilds. Each guild, because all the members in the same guild increase or decrease at the same time, so we can lump them together, generate guild level abundance. Now we have only 18 variables. Then we try to correlate the guild level abundance with all the clinical parameters we collected. Interestingly, you have 3 guilds. They show negative correlation with most of the disease phenotypes. And 9 guilds, they show positive correlation with the disease phenotypes. And 6 guilds showed no correlation. So now we have mapped 161 genomes, which still account for nearly 60% of the total sequence we generate. So even though only 161 bacterial genomes we focus on, is still a good part of the data we originally generated. They are still prevalent and the predominant members of the gut microbiota. And now we have their interactions between each other, so they actually form 18 guilds. But we also have their relationship with the host. The 3 blue arrowed ones promoted by the new diet, and they show negative correlation with disease, and they are potentially beneficial to the host. The 9 red arrowed ones, they are reduced by the diet, and they show positive correlation with disease, and they are potentially detrimental or pathogenic to the host. And you have other guilds showed no correlation with disease, and they are potentially neutral. So this work introduced the guild concept into human microbiome field. And so a guild is potentially a functional group formed by different members in a gut ecosystem. Hello?
Yeah, we can hear you.
I heard some people speak on the background, and
yeah, please.
Sorry.
No.
Okay, so I thought it was you telling me something.
No.
Okay, so basically, if you look at the members in a guild we identified in this particular study, they can be from widely different taxonomic background. For example, this particular guild had members from 4 different phyla. So bacteria, they behave in an ecological way. They cooperate with each other regardless of the taxonomic background. And so this is one thing we need to keep in mind. The other thing is, even in the same species, we can often assemble and identify several genomes. For example, we have 5 genomes in this butyrate producing species, Eubacterium elegans, and you can see they actually behave in 3 different ways. So some are reduced by the intervention, some are slightly promoted, some showed no change. But if you lump all the 5 together, this is what you see. It's actually artificial trend. It's never exists in an ecosystem. So that's why we should not, bacteria work as guilds, not as taxa. So if you would like to, it's okay to do a survey of the microbiota based on taxonomy, but you should not correlate any taxonomic level data with the disease, because even in the species, you have opposite changing members. So you should not lump them together and to do correlation with disease. This is one study on obesity, but the clinical design is self-controlled. So we know self-controlled design cannot demonstrate causality much. So all the data I presented to you is just showing that probably can convince you that bacteria, different bacteria can form guild and respond in a guild level structure. No more than that. But now I would like to show you another study. So it's a high fiber dietary intervention on type 2 diabetes. We try to more rigorously answer the causality question. So basically, we we randomize type 2 diabetes patients into two groups, and the Eurocare group and the high fiber diet group. Because we cannot remove all the drugs, so we gave the same drug to both groups. It's the drug called acarbose. It's an alpha amylase inhibitor, which can reduce starch digestion, so that can reduce the glycemic response, but actually make the undigested starch available become dietary fiber. So both control group and the treatment group, they had a basal level of starch as the dietary fiber in their gut microbiota. However, we controlled the total calorie intake and the macronutrients intake between the two groups, but added about 40 grams of very diverse fiber in the treatment group. And now you can see after 3 months of intervention, both groups showed a reduction of HbA1c, but the treatment group showed even further reduction. So there's an extra clinical benefit due to the extra intake of dietary fibers in the treatment group. Because of the randomized design, controlled design, you can only attribute the extra clinical benefit to the extra intake of dietary fibers. And the only way for dietary fibers to work to change, to improve type 2 diabetes probably would be by way of changing the gut microbiota. So again, we transplanted the microbiota from the same patients at the baseline and 3 months after. And the baseline microbiota induced high glycemic response in the recipient mice. So that means the diseased gut microbiota from type 2 diabetes patients can actually increase fasting blood glucose level and also OGTT response of the tolerance, glucose tolerance to the intolerance to the recipient mice. But the after intervention microbiota from the same individual didn't have such adverse effect on the recipient mice. So this is the indication that the dietary fiber is the causative factor for improving type 2 diabetes, and it's by way of changing the gut microbiota. And then we did metagenomic sequencing of all the 172 fecal samples, and the same strategy as in the PWS study. We end up with nearly 500 million non-redundant microbial genes. And we did co-abundant gene analysis. We identified 422 bacterial genomes. And by using these 422 bacterial genomes to look at the overall structural changes of the gut microbiota, we can say that at the baseline, the two groups showed no difference. But after 1 month of intervention, after both treatment group and the control group, they become different from their baseline. And they also become different from each other. And after the 1 month, and at 2 months and 3 months, if you analyze the microbiota, they become they're stable and no further change. So this is indication that when you include high amount of dietary fibers in a patient's diet, it took about 1 month to stabilize a new structure of the gut microbiota. If you keep the diet, you keep the new structure, and at least for the 3 months period. We also analyzed the total variations of clinical data and the total variations of the gut microbiota, and did a correlation between these two, and showed that the more you change your gut microbiota, the better clinical outcome you may achieve. So there is a correlation between these two. And we know that dietary fibers are innate to us. So, but gut bacteria can metabolize, ferment them into short-chain fatty acids, and short-chain fatty acids may have bioactive effects. So we look at the variations of 6 short-chain fatty acids throughout the trial in the 2 groups. We found 4 of them actually reduced after intervention in both groups. Then they cannot be responsible for the improved disease phenotypes. And then we focus on acetate and butyrate. So acetic acid showed no change and also no difference between the two groups. It's potentially maybe beneficial or responsible for the commonly shared clinical benefit of the two groups. But the butyric acid showed no change in the control group, but significantly increased in the treatment group. So this butyric acid change may be responsible or mediating the treatment group only clinical benefit. How short-chain fatty acids, acetic acid, butyric acid can actually help type 2 diabetes patients. There are already many studies showed that they can bind to GPCR41, 43 receptor in an L cell in our gut and stimulate GLP-1 production. When they produce more GLP-1, the gut peptide hormone, and that hormone actually can stimulate pancreas beta cell to produce more insulin. If you have higher insulin production, you have better glycemic control. This is probably one of the potential mechanisms. And however, we also see the drop of pH of the fecal matter. So this is indication that the gut has become acidified. And as a microbiologist, we know that pH is an environmental factor which can change the growth behavior of almost everybody in the ecosystem. So we need to look at the growth behavior change of the major members of the gut microbiota. From the 422 genomes we identified, 180 actually shared by 20% of the samples. So they are prevalent bacteria. And we assembled 154 high-quality draft genomes from these 180 co-abundant gene groups. And we did annotation of their gene functions. 141 of such genome harbored at least one of the key genes for short-chain fatty acid production. So this is an indication that all these 141 high-quality draft genomes of prevalent gut bacteria, and they are potentially capable of fermenting some kind of dietary fiber and produce one of the short-chain fatty acids. So they are genetically capable of using the available dietary fiber and produce short-chain fatty acids. However, if we look at their response to the diet, only 15 eventually become predominant, and they increase their abundance and become predominant. And 79 actually showed no change. And 47 even actually reduced their abundance number from baseline. This is a surprise to us because at the beginning, we try to provide such a diverse range of structures of dietary fibers with a high amount in hoping that all the short-chain fatty producing bacteria would find something to grow on. So we promote the diversity of such bacteria, maybe that's beneficial, but it's the opposite. Instead of promoting the diversity of the general short-chain fatty acid producing bacteria, we only selectively promoted a small group of short-chain fatty acid producing bacteria. Large group of them do not respond to the high intake of diverse dietary fibers, and quite a few of them actually reduced by the high intake of dietary fibers. So these 15 bacteria, each of them promoted by the dietary fibers, they are potent responders to the dietary intervention, and they all can produce acetic acid. 5 of them can also produce butyric acid. And if you look at the 47 negative responders, some of them can produce indole. Some of them can produce hydrogen sulfide. Many of them can produce endotoxin. So they are potentially detrimental or pathogenic. And there's a negative correlation between the positive responders and the negative responders. But is this just a negative correlation, or is there any possible or plausible causative mechanism between the rise of these 15 and the decline of these 47? So we look at the genomes of these positive responders. We found that they can produce more ATP potentially from the same amount of carbohydrates they fermented compared with many other bacteria. And they can also produce more short-chain fatty acids. And they are also tolerant to low pH. So they grow quicker and they acidify the gut environment quicker but they are tolerant to the acidified gut environment. So even the pH reduced lower than 5, they can still continue to grow. If you look at the negative responders, it's the opposite. They get less energy from the same amount of carbohydrates. They are not tolerant to low pH. So probably at the beginning, everybody start to grow by taking the advantage of the new energy source, but very quickly, you start to see difference. So this group would continue to grow. This group quickly stop grow and then even decline. And so there is a plausibility that the increase of these 15 and actually lead to decline of these 47 potentially pathogenic bacteria. And so now we look at the genomes and compare them with the database. We found some of them are very close to what's already known species in the gut microbiota, beneficial species. Some can only be identified at the genus level, some at the order level, family level. So some are potentially, some are novel, some are well-known species, but altogether they form a guild. They are from 3 different phyla. They work together, promoted jointly by the new fiber diet. And if you develop an index combining the diversity and total abundance of these bacteria in each individual human gut microbiota, and you can see there is a positive correlation between the rise of the abundance and the diversity of these positive responders and the decline, the reduction of HbA1c. And so basically, this is a study to show that in addition to the direct beneficial effects for producing acetic acid and butyric acid, increase GLP-1, increase insulin, that's a direct molecular crosstalk between these positive responders who can produce acetic acid and butyric acid. There's another ecological mechanism for them to be beneficial. They actually acidify the gut. They also produce antimicrobials. Particularly butyric acid itself is actually antimicrobial. So if you reduce the pH by using hydrogen chloride to the same level, you don't see the same level of inhibition to pathogens. You actually need to use butyric acid to reduce, acetic acid to reduce the pH to the same level. You see much stronger inhibition. And because they grow, they occupy most of the available niches, so they also drive the pathogens down. So all these ecological effects together drive the endo producer, hydrogen sulfide producer, endotoxin producer to a very low level. Endo and hydrogen sulfide can inhibit L cell from production of GLP-1. Endotoxin can induce inflammation. Inflammation can damage insulin receptor, increase insulin resistance. So after you reduce endo producers, hydrogen sulfide producers, endotoxin producers, you release the inhibition to L cell production of GLP-1. You reduce inflammation. You recover insulin sensitivity. That's another aspect for how this group of bacteria may be beneficial to type 2 diabetes patients. And we also start to isolate specific members from this positive bacteria group, positive responders. And now we have this one, Bifidobacterium pseudocannulatum. It showed protective effect on high-fat diet-induced obesity in mice. And it also reduced the glycemic response in mice inoculated with microbiota from type 2 diabetes patients. So basically, these are also causative evidence to show that members of this positive responder guild should be very important to patient health recovery. So now our hypothesis is that this particular group of positive responders to high fiber diet intervention may be a foundation guild, because they have very different species. So we call and work as a guild. Instead of calling them foundation species like the tall tree species in close forest, we call them foundation guild. And they are the foundation for restructuring and restoring a healthy gut microbiota. And each individual may have different bacteria to form, to become the members of the foundation guild. And the different member actually require different fermentable fiber and as energy to support their growth. So this comes a new avenue for personalized nutrition by targeting this foundation guild with the fermentable dietary fiber combination. And some patients, there's data already shown that some patients may have lost their original foundation guild, and so they need to be re-inoculated and to be able to recover their healthy gut microbiota. And so in summary, I would like to again emphasize the 4 important aspects for designing, conducting, and analyzing a microbiome study. First, keep causality question always in mind. And in your study design, you should always strengthen the possibility that your data can show a piece of causality evidence. Second, try to analyze your data down to the string level as much as you can. And also keep in mind that bacteria work together as guilds, naturally, ecologically, but they don't work together as a genus or as a family or as a phyla. And this is also very important for you to understand your data. Lastly, but not least importantly, do not use the existing database to initiate your microbiome analysis. Try to use a pipeline which require no prior knowledge. And only after you identify some potentially important bacteria, and then compare your sequence with the database, so you'll know which part of your finding is novel, which part of your finding has already been touched upon by other people before. And 1 more minute, I would like to comment that there are already many animal studies to show that short-chain fatty acids can also boost both innate immunity and adaptive immunity for antiviral reaction. And there are so many studies out there already. So potentially by recovering the foundation guild for producing short-chain fatty acids, we may also help COVID-19 patients or any other viral infection patients regain their antiviral immunity. So this is a possibility there. And by reducing there's also data to show that during infection, viral infection or bacterial lung infection, when people develop a fever, there is a surge, there's a spike of opportunistic pathogens during fever in the gut microbiota. So if you can maintain a healthy gut microbiota, keep the opportunistic pathogens at bay without a spike, you may actually reduce both the cytokine storm due to huge amount of endotoxin released from overgrowing pathogenic bacteria, or from sepsis due to potentially translocation of such pathogenic bacteria across gut barrier to internal organs. So I would like to thank all my students and colleagues and collaborators and the funding agency, and I'm happy to answer questions.
Thank you very much. Thank you very much, Liping. That was great. Great talk. I will try to get, I apologize to everybody. I'm really not used to webinars, so this is why I may. So I'm going to try, and Jerry, I see the, oh yeah.
You see the question from Dr. Grower?
Yeah, I saw it. Maureen, you said, maybe Maureen, you can speak, I mean.
Okay, I'll unmute her.
Maureen, can you unmute and speak, or do you want me? I see a question from Maureen.
Yeah, let me, okay, she should be able to speak.
I'm able to speak, I think.
Okay,
yep.
Please.
And I think it was probably a question that was answered in a later slide. But when you were looking at the positive guild and the reduction of members and the production of acetate and butyrate, I wondered if it was largely coming from bifido or actinobacteria and firmicutes. But then in the next slide, you had some information about that. And I think I was being maybe too reductionistic in my question.
Okay. Well, actually, if you look at the 15 bacteria identified in this particular study, they are from 3 phyla, Firmicutes, Actinobacteria mainly, these 2 phyla. Only 1 from Proteobacteria. No members from Bacteroidetes, and very, very small, no, only 1 from Proteobacteria. And this is a surprise to us at the beginning. And now we understand why. So because many studies on dietary fiber and gut microbiota are actually used members from Bacteroidetes. And the most well-known like Bacteroides, Cytotomicron. And however, they are sensitive to low pH. So they have a very versatile capacity for fermenting all kinds of fibers, but only at a pH near neutral. When the pH reduces below 5.5, for example, almost nobody from, no member from that phyla can grow. So that's probably why. And we had other studies which are unpublished and showed the same trend. No members from the Bacteroides phyla can survive such a low pH, so they don't show up there.
Thank you.
Thank you. I have a question. I mean, maybe to.
Okay. Yes, I'll wipe down the box. I'll wipe down the box. Are you going to be ready to go?
No, somebody should be on mute, I believe.
All right, I'll wipe down the mask.
Somebody should be on mute. Okay? That's always the problem with the webinars or these conference calls. So, okay, so I just want to ask Liping, I mean, this is a general question. First, I really believe, and this is very clear from his talk, but I really want to draw again the attention of all the attendees on the fact that the concept that Liping is bringing up with the Foundation Guild as against works only based on taxonomic of a species is really completely overhauls the way we can design studies. And I believe that this is something we may have the possibility to discuss in the future for all what is going on at USF. But the question is, do you have the possibility, or maybe we can help on this, and do you think it would be interesting to really compare these foundation guilds between in different geographical areas? I mean, what would happen, for example, if we were investigating the same study, for example, in obese and diabetic patients in Africa, for example, or in South America? Do you have any clues to this?
Yes, and this is a fundamentally important question. Well, if this is a foundation guild, and that means they need to be also the pioneering species for newborn gut microbiota. And we know that newborn babies have a naive immune system, and their gut is actually also pH neutral and rich in nutrients. So it's actually first come, first colonize situation. So if you colonize the newborn's gut with some opportunistic pathogen, and then it's a trouble. But if you colonize with these foundation guild members, it's protective. So that's why our hypothesis is that this kind of bacteria, because they are so essential to protect us from suppressing pathogens, so they need to be in newborn baby's gut from the very beginning. And so then, so they need to be vertically transmitted from one generation to the next. And we now know that the best mechanism is through mother-infant transmission. First, members from the reproductive tract, they can be members of the foundation guild, and bacteria in breast milk also can be foundation guild members, and skin of the mother And and also the breast milk contains 10 to 15 grams of oligosaccharides naturally produced, 200 kinds, and this is already well known, and to promote the growth of such bacteria. So that's why naturally delivered breastfed baby, their gut microbiota actually overgrown by members of Foundation Guild, by such bacteria, similar bacteria. So then if these bacteria are co-evolving with us and vertically transmitted, then they should have the ethnic, so now we think ethnic background, geographic location, dietary pattern, and gender are probably 4 major factors which affect the composition of the Foundation Guild. And so, yeah, if you do the same study in different geographical locations, you may find, and with different genetic background, you may find different people actually have different members of gut microbiota as their Foundation Guild. And of course, just like the trees, also you have different trees. Some tree can grow up to 100 meters, some tree can only grow 10 meters. So even though you have a forest, some are very complex because you have a really tall tree. I think Foundation Guild bacteria also like that. Some are very strong, very good. Some are not so good, but still okay. If you keep them, carefully keep them, they can still keep you healthy.
Thank you. I believe that Shyam, you have a question.
Let me.
I can read the question here. Maybe you don't need to unmute all the time. So I have one question. Breast milk oligosaccharides foster Foundation Guild? Yeah, the answer is probably yes. But this is a hypothesis, and we need to work on that. Is there any gut microbiome difference between soluble, insoluble fiber consumption? And the answer is generally yes. And it's actually, the question is not soluble or insoluble. The key is fermentable or non-fermentable. So dietary fibers, chemically, they can be divided into fermentable dietary fiber and non-fermentable. Some are just chemically cannot be fermented by any bacteria. But among the fermentable fibers, to different person, this structure may be fermentable by my gut microbiota or my Foundation Guild members, but not by yours. This is where we need go into personalized nutrition.
Thank you. I see another question. Is there, do you see it?
Yeah, is there any gut microbiota difference between soluble, insoluble fiber consumption? Yeah.
Shyam, do you have questions?
Let me unmute him for one.
Yeah. Shyam? I'm not sure that he gets. Oh.
Okay, I have another question coming in. Can you expand, clarify your statement? Do not use a reference database for your microbiome analysis. I see, more probably, I say do not start your microbiome analysis with a reference database. This is what I mean. So we know currently when you generate 16S RNA sequence or metagenomic sequence, after you do quality filtration and sort out, you generate your ASV table or your non-redundant gene microbial gene catalog. That's where you need to start your data analysis. But many people start, the next step is to compare your own sequence with an existing database so that you can either assign a taxonomic name or you can assign a gene function to your own sequence. The problem of this approach is that a large chunk of your data, usually 30%, sometimes even over 50% of the data, cannot find a nearest neighbor in the existing database. So you cannot give a name, you cannot give a function. What do? You just leave them aside. And you just analyze those sequences which have a nearest neighbor, which can be given a name or can be given a function. This is a problem because you always look at the part of your data which have already been studied by other people before. The novel part of your data, you don't. And this is why I say, please do not start your microbiome analysis by using a reference database. And actually each sequence, no matter it's 16S or it's a metagenomic gene sequence, each unique sequence represents a unique function or a unique bacteria. So that just look at their behavior and identify the sequence as a tag. After you identify the potentially important sequence tags, then you need to go to compare your sequence with the database so that you know which part of your finding is novel, which part of your finding is actually connected with reported case before.
Okay. Thank you. Do we have I always see Shyam, but yeah, please.
Okay, so first of all, thank you, Liping. I think that's an excellent webinar. And we have about 50 people joining this webinar. So this has been fantastic. And we're trying this out with you first. So,
thank you.
Now, I had two questions. One was how long it takes for the big guild to establish. Now, I know your studies were in 3 months, but how long really it takes? Have you looked at that time points?
Yes, and we actually have data. We publish the data at a monthly interval. So from baseline, 1 month. And then you can see after 1 month, the new guild formed and become stabilized. But we also have data 3 days after the new diet and 1 week after the new diet. You see they are increasing. Even at 3 days, they are already significantly higher than the baseline. But they reach plateau after about 4 weeks. And we also have data to show that after 3 months of a high fiber intervention, we stop the intervention. They go back to their normal daily diet. After 1 year, we get them back, and they have their disease back. They have their microbiome back. So okay. You need the nutrition. The new nutrition support the new microbiome and support the new health. If you remove the nutrition after probably also a few weeks, you go back to your, so one dietary pattern supports one structure of the microbiome and that structure has its own health implication.
Okay. I believe that Joseph, you had a question, Joseph Dassault, or maybe I'm wrong.
I'll unmute him real quick. Let's see.
If you want, you can also just write your question in the messages, but I saw that, Joseph?
Okay.
Maybe not, maybe that was not a question. No.
Okay.
So do we have final questions?
I have a question.
Oh, please.
I was wondering, if your hypothesis is correct and you're serious about reducing the risk of the COVID-19 pandemic, have you considered having a group where you would just modify the diet, such as a Mediterranean diet, and then also another group where you would have someone with a group with the Mediterranean diet and taking your fiber supplement? Because if one eats a poor diet, like a high-fat, high-sugar diet, that would counter the effects of your fiber supplement.
Yes, actually, we, Dr. Shaw and we are organizing a COVID-19 project exactly focusing on that question. So if we can use dietary fiber supplement, we need to provide fermentable fiber up to 40 to 60 grams a day. That's a huge amount to ensure that the whole long gut, everywhere you have extra dietary fiber to ferment. So it's not that only the upper part of your colon has enough fiber to ferment and suppress the pathogen, but after the carbohydrates used up, the lower part is still a dysbiotic gut microbiota. you need to ensure that the whole gut, the whole colon actually have enough dietary fiber to ferment to support the foundation gill, not just the first part. So but anyway, we are organizing a clinical trial, 3-center clinical trial, Rutgers University, USF, and Columbia, and we are just pushing for that forward. Yeah.
Thank you. We have a question from Maureen. She has written the question if you want to read it.
Okay. I work with children preterm with marked dysbiosis and delayed succession. Any idea about how to manipulate the diet and breast milk effects swamped by gamma proteobacteria? Yeah, I agree, but this is not my area, so I don't have data. But potentially,
be ready, John.
They are.
Please mute.
Somebody yelling, and I don't know. So, but anyway, I think these two questions are very important, and if you probably need to reintroduce members of the foundation guild to these children's gut, and using breast milk to support their growth, and rebalance the gut microbiota. So we know that they are dominated by, their gut microbiota usually dominated by gamma proteobacteria and very nasty pathogens. And so we need to introduce the foundation guild, probably. So this is also a hypothesis. And we have another question. After colonoscopy, when the gut is clean, are there any recommendations for reestablishing the gut microbiome? It might be a good idea to start a new diet after your gut cleansing, so that the new diet may take a shorter time to establish your new microbiome. Yeah.
Yeah, these are important questions indeed.
Yeah.
Okay. Well, thank you so much, Liping. That has been great. Okay. First webinar of the club on microbiota. This is about history. And thank you to all of you for attending. And again, Liping, and this is extraordinary, is really connected to USF, working as a consultant. As he mentioned, we are organizing some joint clinical trials. So really do not hesitate to share ideas, to share projects, and that would be obviously fun and very interesting. Have a very good day, all of you. Be safe, and we will organize a follow-up of this webinar, of these webinars. Thank you, Liping. Bye-bye.
Thank you. Bye-bye.
Bye-bye. Bye, everyone.
Bye. Bye, everyone.
Bye, everybody.